Our lab has been working for a number of years on a protein project that really is an enzyme, and an enzyme is a protein that does a particular reaction. This reaction and this protein is kind of weird in a way, and the real point I want to get across to this is not to tell you the story about a weird protein and some interesting activity that we discovered from it, but it's to point out the differences between doing fundamental science, doing foundational science, basic science, science of things that we just don't understand and we really wish we did, and doing science which is more translational, where there's an end goal that has something to do with business or medicine or human health or something like that. So the protein we look at is an endonuclease, and it's really important in a lot of industrial applications. And in fact, our original work with this enzyme started out because of a collaboration with a company. They had noticed that this enzyme that they're trying to sell has these weird behaviors, and we thought, well, the weird behaviors are cool. One of the things that it does is that it binds a certain sequence of DNA, specific sequence of DNA, and it cuts that sequence. So it's an endonuclease. It cleaves nucleic acids, double-stranded DNA in this case. So in certain circumstances, the activity actually accelerates. It gets faster, and usually it gets much, much faster when there's more of the primary sites, as they're called, the canonical sequence that it normally binds and cuts. And if there's too many of those sites around, it turns out that the enzyme actually starts cleaving other sites, which is an off activity, something that the company didn't want to have, and they wanted to figure out why. Their original hypothesis was that this enzyme would recognize a primary site, cut it, cleave it, and it would have its conformation somehow changed such that it would then be more prevalent to binding to secondary sites and cutting things that it's not supposed to cut. And they wanted to figure out how that conformational change happened. And it turns out that's not what the enzyme does at all. It's actually kind of more interesting and more cool in a lot of ways. The enzyme forms fibers of the primary site bound to DNA, the primary DNA site bound to the enzyme, and fibers of this complex grow and change the structure so that the secondary site, which generally binds the enzyme but doesn't ever get cut, when the secondary site is bound into a fiber form of the enzyme, then it does form. It does get cleaved. And so this whole process, this whole journey of discovery was really interesting. And along the way, the company heard everything they wanted to hear about this, and the rest of us that work in the lab thought that this was still an exciting project because we wanted to understand how an enzyme and why an enzyme would evolve the ability to form fibers. What sort of purpose does this give the enzyme? And it turns out there's a whole host phage arms race between bacteria and the viruses that infect them, and this enzyme happens to be part of what is an important immune response for this particular bacteria for fighting off viral infections. The virus happens to have these secondary sites fairly prevalent in its genome, so the enzyme supposedly binds the primary sites, cleaves the primary sites, and at the same time starts cleaving the secondary sites, but only of the invading phage, only of the invading virus, and not of the host genome of the bacteria. This is kind of a neat solution to a very difficult problem for a bacteria that's under a viral attack. But the point I want to get across is that this is not always why we do science. It's not always to come up with some answer for a company. One of the reasons we do science is because we just want to figure out how and why certain things work. There's no end goal. There's no monetary gain for this. There's no obvious end application. But what's really interesting is that if you look into it deep enough, there's plenty of people that are engineers and scientists and clinical scientists that are really good at taking discoveries like what we've worked on and turning it into applications that are useful. So with that, I think I'll just end. Thanks.